Hello and welcome. This is Columbia Basin Research's 7th video in a multi-part series on using Program SURPH. Program SURPH is a program created by Columbia Basin Research at the University of Washington School of Aquatic and Fishery Science. Program SURPH, Survival Under Proportional Hazards, was developed to analyze data from release-recapture studies of animal populations, in particular to relate the survival estimates from release-recapture studies to individual-based and group-based covariates. Today in this 7th video, we will demonstrate the relationships of migration distance to survival as well as link to survival. Alright, so let's fire up the program. This example will illustrate modeling survival as a function of a group covariate and using the group survival plot to visualize the modeled survival probabilities as a function of a group covariate. The data for this example is in the distance dataset. Here we can see that the first three populations traveled 116 kilometers to the first detection point, dam, the fourth 176 kilometers, and so on. We can perform two-stage modeling and begin with modeling the detection probabilities as was done in the previous video. The result of this will be to select a full model with population interval-specific effects for the detection model. There's no reason to believe that the migration distance will have any effect on the detection probabilities. Once the full detection model is selected, I can double-click on AnnoDev and select the Use Defaults option. The Analysis of Deviance dialog will appear. A p-value of 0.000 for the full covariate model indicates the migration distance has a very significant effect on survival. It might be a reasonable assumption that it would affect survival for the first interval but not the second. We can test this by creating a new model, distance 1, that models only the first interval survival as a function of distance. Make sure that the model underscore full covariate is the active model and open the group effects dialog for survival. I can then set it to this configuration. Create the survival model, naming it distance 1. After I double-click estimate, the new model will show up in the Analysis of Deviance dialog. I can then add it to the AnnoDev table by selecting it and pressing the down arrow. The result is shown here. A p-value of 0.000 for distance 1 versus underscore grand mean shows that the migration distance has a very significant effect on survival in the first reach. A p-value of 0.126 for underscore full covariate versus distance 1 shows that the migration distance does not have a significant effect on survival in the second reach. We can use the group survival plot to visualize the effect distance has on survival. Make sure that the preferred result, distance/distance1, is the active result, and double-click on Group under Survival Plots, under View Results in the Results section. It brings up this plot. The x-axis is the migration distance and the y-axis is the survival probability. The pluses on the graph are the survival estimates for each of the 10 populations using the CJS model. The data point for population 10 is here. The red curve is the modeled survival probability as a function of covariate. The way that the model survival curve fits the CJS estimates indicates that the model fits the data adequately. A further example can illustrate the process of modeling survival as a function of an individual covariate. In 1993, a study was conducted in the Snake River to study the relationship of body length to survival. A total of 1,405 fish were captured, their lengths were recorded, and they were pit tagged and released above the Lower Granite Dam. The file reservoir contains the data for this study, and the data with individual covariate data are loaded into SURPH, and individual covariate graph is provided for exploring the individual covariate data. The Kolmogorov-Smirnov KS test shows a p-value of 0.0858, which is marginally significant, showing the individuals known to be alive in interval 1 may be larger than those not known to be alive. This may be because length affects the detection process and/or the survival process. Using the two-stage modeling approach will allow us to first model detection as a function of length independent of its effect on survival. Once the detection modeling is done, we can then model survival as a function of length. Modeling the detection probabilities first will prevent the effects of length on detection from being mistakenly attributed to the effects of survival. Because there is only one population in this study, there are only two parameterizations for the detection main effects, a unique detection probability for all intervals or a common detection probability. The models are created by using the main effects dialog to set the parameterization as has been described in previous examples and creating the models using Create Model under Detection Only Modeling. The interval-specific model is named Full Detection, and the model with the common detection probabilities is named Common Detection. After estimating the parameters, we can bring up the likelihood ratio test dialog. When comparing the full model with the common model, the p-value is 0.0089, indicating that the full model is a much better fit. We next explore whether length has a significant effect on detection by opening the individual effects for detection. Ensuring that the full detection model is the active model before proceeding, you can create two models. Number one, the model parameterized as shown here that assumes assumes that the length affects detection in the same way in both intervals. And number 2, the model parameterized as shown here that assumes the length affects detection differently in each interval. Call the first model common length det and the second unique length det. We then estimate the parameters and return to the likelihood ratio test. Testing either model against the full detection model fails to show a significant p-value. We thus conclude that length has no significant effect on detection and conclude that the full detection model is the best model for the detection probabilities. We thus lock the model and proceed to the modeling of the survival probabilities. As with the detection probabilities, having one population allows for only 2 possibilities for modeling the main effects. First, set the survival main effects to interval specific and create a survival model named full. Then set the main effects to common and create the survival model named common. We can estimate the parameters and use the likelihood ratio test to compare the models. The p-value indicates the full model is no better than the common model. We can thus use the common model as the base for modeling the individual covariate effects. Ensuring that the common model is active, we can model the survival individual covariate effects as we did for the detection probabilities. We can create a model called common length that assumes the length affects survival the same way in each interval, and another model called unique length that assumes a unique length parameter for each interval. We estimate the parameters and return to the likelihood ratio test dialog. The likelihood ratio test dialog with the new results is shown here. Comparing the common length result with the common result yields a p-value of 0.0581, indicating a significant result at the 0.10 level. And comparing the unique level result to either the common result or the common length result yields an insignificant p-value, indicating that the model that uses a common effect on length on both intervals is the best model. Making sure that the reservoir common length result is the active result, we can look at the individual covariate survival plot for visual representation of survival as a function of length. Double-clicking on individual under survival plots under view results brings up the individual covariate plot. The smooth line represents survival modeled as a function of the length for interval 1, as defined by the currently selected model, and the more jagged line is a non-parametric estimate of survival as a function of length. The parametric estimate appears to conform well with the non-parametric estimates for length, less than about 150 millimeters, but does not conform well for larger individuals. If we select Display Histogram, a histogram of individuals at risk in interval 1 is superimposed on the plot. Here we can see that where most of the individuals' lengths are, the parametric function conforms well to the non-parametric function. However, where the parametric function conforms less well, there are very few individuals. Here, we're able to see the same plot for survival in interval 2. In this case, the fit of the parametric function to the non-parametric is not as good. More modeling is therefore indicated. This has been Lesson 7 on using Program SURPH, in which we covered the relationship of migration distance to survival as well as length to survival. This is the final video in the SURPH series. Please note that if for any reason you are having trouble with Program SURPH and these videos are not sufficient to answering your questions, you can find the user manual at cbr.washington.edu/analysis/apps/surph. The PDF manual is there, and you can either load that into the browser as I'm doing right now, or you can download it to view in Adobe. If you're using Program SURPH and you have any questions or comments on that same page, you may also give us feedback via our user satisfaction survey. We would greatly appreciate any feedback you have in helping us make future versions of this program to be more useful to our users. We hope that this video helps you get the most out of Program SURPH. Again, if you have any questions or comments in regard to Program SURPH, please fill out our survey as previously detailed. You can also contact Columbia Basin Research at web@cbr.washington.edu. Best of luck on your analyses, and thank you.